Nucleus. Before going into details of nucleus structure and function, let's discuss few points about cell organelles. Organelles are defined as membrane-enclosed little organs suspended in the cell cytoplasm. Each organelle has a definite structural makeup and plays a critical role in cellular function. Many different cellular organelles function together to keep the cell healthy. Cell organelles are classified into three categories depending upon the presence or absence of membrane and number of membranes. Amembranous, single membrane-bound, and double membrane-bound organelles. Amembranous organelles, single membrane-bound organelles, and double membrane-bound organelles. Cell organelles are not bounded by any membrane. These organelles are bounded by a single membrane. These organelles are bounded by a double membrane. Amembranous organelles are present in both prokaryotes and eukaryotes, whereas single membrane-bound and double membrane-bound organelles are found only in eukaryotic organisms. Examples include 70S and 80S ribosomes, Nucleolus, microtubules, centrioles, flagella, and cilia. Examples of single membrane-bound organelles include rough and smooth endoplasmic reticulum, Golgi apparatus, vacuole, lysosomes, microbodies like peroxisomes and glyoxysomes. Examples of double membrane-bound organelles include nucleus, mitochondria, and chloroplast. Now, let's discuss the structure and functions of nucleus. Nucleus was discovered by Robert Brown in the year 1833. It is a membrane-enclosed organelle located in the center of the cell. DNA-protein complex present in the form of chromatin threads is found within the nucleus. Nucleus is the primary site of gene expression and DNA replication during cell cycle. It is perhaps the most critical organelle as it is the cell's command center which controls all of the cell's activities, such as growth and metabolism, using the DNA's genetic information. All eukaryotic cells contain nucleus except few cells, such as erythrocytes, some lens cells, and mature sieve tube cells of phloem. Prokaryotic cells lack nucleus. Instead, the cell's DNA is distributed loosely around the cytoplasm. Uninucleate cells. Usually, cells contain only one nucleus. Examples: smooth muscle cells and cardiac muscle cells. Binucleate cells. Some cells contain two nuclei. Example: paramecium. Macronucleus with somatic functions. Micronucleus with reproductive functions. Multinucleate cells. Some cells contain more than two nuclei. Examples: syncytial skeletal muscle cells in animals. Cenocytic tapetum cells in plants. Morphology of the nucleus. It is a double-membraned organelle. This is the outer membrane, inner membrane. Between the outer and inner membranes is the intermembrane space or paranuclear space, nucleopore complex, and protein network called nuclear lamina. These all components together constitute the nuclear envelope. This dense body within the nucleus is the nucleolus, and these are chromatin threads carrying the genetic material, DNA. The gel-like fluid inside the nucleus is called the nucleoplasm. This is the rough endoplasmic reticulum studded with ribosomes, and is continuous with outer nuclear membrane. This is the cell cytoplasm. Going into details of each component of the nucleus. Firstly, nuclear envelope. Nuclear membranes. The nucleus is enclosed by a double-layered nuclear membrane. Outer membrane is continuous with the rough endoplasmic reticulum. Inner membrane contains inner nuclear membrane proteins such as emerin, lumen B receptor. Intermembrane space, also known as paranuclear space, is the space between the inner and outer nuclear membranes of the nuclear envelope. The nuclear membrane serves to protect the genetic material in the cell, and partitions the chromosomes from the cytoplasm and other cellular contents. Nuclear lamina. It is a protein network present beneath the inner nuclear membrane. Lamina is composed of proteins called nuclear lamins A, B, and C. Any mutations in nuclear lamins lead to diseases called laminopathies. For example, mutation in LMNA gene leads to Hutchinson-Gilford progeria syndrome. Functions of nuclear lamina: it provides mechanical support to nuclear membranes. It attaches to and organizes chromatin. Nuclear pore complexes, abbreviated as NPCs. Nucleopores are an array of tiny pores that are present within the nuclear membrane. Nucleopore complexes are gateways of nuclear import and nuclear export. NPCs contain a central channel and octagonal symmetry. These pores allow the selective passage of molecules such as proteins and nucleic acids between the nucleus and cytoplasm. Mode of transport and molecules transported. Facilitated diffusion of ions, metabolites, small proteins less than 40 kD, and active transport of large proteins, mRNAs, tRNAs, and ribosomal subunits. Nuclear import and export requires two components: karyophorins, and RAND, which is a small G protein. Karyophorins are categorized into two types based on their functions: nuclear import receptor called importin, and nuclear export receptor called exportin. RAN is a GTP-ACE, which binds guanine nucleotides, that is GDP and GTP. Nuclear import. It requires two components: importin and RAN. Nucleus imports proteins such as histones, DNA polymerase, RNA polymerase, transcription factors, replication proteins, and repair proteins, etc. Nuclear proteins are synthesized on free ribosomes, and are targeted in post-translational matter. Proteins targeted to nucleus contain an address called nuclear localization signal. NLS is rich in basic amino acids. Nuclear localization signal is either monopartite NLS or bipartite NLS. Monopartite NLS contains a single cluster of basic amino acids. It was first discovered in large T antigen of SV40 virus. Bipartite NLS contains two clusters of basic amino acids. It was first discovered in Nucleoplasm. Nuclear export. It requires two components: exportin and RAN. Nucleus exports mRNA, tRNA, and preribosomal subunits. The rRNAs are combined with proteins to form ribonucleoproteins. The proteins contain an address called nuclear export signal. One type of nuclear export signal is rich in leucine. Let's dive into the details of nuclear import and nuclear export diagrammatically represented here. Primarily nuclear import. In the cytoplasm, cargo protein containing nuclear localization signal forms a complex with importin. This bimolecular complex enters the nucleus through the nucleopore complex, which is a large protein assembly that is embedded in the nuclear envelope. NPC is composed of cytoplasmic fibrils and nuclear basket. Bimolecular complex is now seen in the nucleoplasm. Cargo protein dissociates from the importin with the aid of RAN-GTP. Nuclear export starts with the formation of trimeric complex in the nucleoplasm. Cargo protein containing nuclear export signal forms complex with exportin and RAN-GTP. This trimeric complex moves to the cytoplasm through the NPC, and thereafter the complex disassembles in the cytoplasm. Cargo protein dissociates from the exportin upon the hydrolysis of RAN-GTP. As discussed in the previous slide, the sequential process that occur in nuclear import and nuclear export are listed here. Nucleoplasm. The nucleoplasm is a viscous liquid present inside the nuclear membrane of the cell. It fills most of the nucleus volume, just like the cytosol of the cell. Functions of nucleoplasm. It stores DNA and enables transcription. Nucleoplasm contains several micron-scaled non-membranous substructures called nuclear bodies, which are categorized into nucleolus, nuclear speckles, promyelocytic leukemia bodies, and Cajal bodies, etc. Nucleolus. Nucleolus was first discovered by Fontana in 1781, and the term nucleolus was coined by Bowman in 1848. The nucleolus is a dense, small spherical subcompartment found within the nucleus. ADS ribosomes, which are composed of proteins and ribosomal RNA, are mainly produced inside the nucleolus. Nucleolus is involved in rRNA synthesis. Only 5 srRNA is transcribed outside nucleolus. rRNA processing. rRNA assembly with proteins to form pre-ribosomal subunits. Nuclear speckles. Also known as splicing speckles or interchromatin granule clusters. These are sites of mRNA splicing factors. PML bodies. Also known as PML oncogenic dots or nuclear domain 10 bodies. These are sites of transcription factors and chromatin remodeling complexes. Cajal bodies. Also called coiled bodies. These are sites of small nuclear ribonucleoproteins assembly. Cajal bodies contributes to the biogenesis of telomerase. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.